Warm greetings. The PCR is a biochemical technology in molecular biology to amplify single or few copies or pieces of DNA, generating thousands of millions of copies of a particular DNA sequence. Developed in 1983 by Carrie Mullis, it is a common and indispensable technique. These are the various PCR machines in our lab. Do's. Make sure all the things are available in your work area. Wear clean gloves and lab coats. In cocktail preparation, prepare the DNTPs freshly. Check the concentration of primer. Check for the presence of magnesium chloride in buffer. Add Taq polymerase at the end of the cocktail mix to prevent the start of the reaction. Make sure there is no exogenous contamination of DNA. The concentration of DNA used must be between 50 and 100 nanogram per microliter. Use new tips for loading each sample. Carry the samples to the PCR machine in ice box. Load the samples one by one. And lock the lid properly. Programming the PCR is the critical part that can affect your results. Set the time for each reaction correctly. Annealing temperature is the most important parameter which depends upon the primer used. The optimum number of cycles is 35. Hold the samples at 4 degrees Celsius after reaction. After the PCR reaction is over, switch off the PCR machine. Unload the samples and place them in ice box. Carry the samples back to lab in ice box. Don'ts. Do not add the ingredients without refrigerated condition. Do not use the same tips again and again without discarding. Do not handle the samples with bare hand. Do not take the PCR tubes outside the ice box while adding the ingredients. Do not carry the samples in hand. Poor primer designing like self-complementary must be avoided. Inhibitors like chloroform, phenol, ethanol must be avoided. Follow the do's and avoid the don'ts. All the best for a successful PCR. Thank you.